All right, good morning, everyone. Good morning. All right. So it is my pleasure to welcome us to the SSB Presidential Address meeting, where we're going to be giving away some awards. And then we'll hear from Dr. Corrie Moreau, who's our past president, who'll be giving the presidential address for SSB. My name is Jessica Ware, and I'm curator and also paper pusher, division chair for the Invertebrate Zoology Division at the American Museum of Natural History. Along with Tracy Heath, who is president-elect, Corrie Moreau, past president, and Elizabeth Jockish, executive vice president for SSB. We've been working really hard this year to try and do some board management strategies, to try and simplify and streamline the way that volunteers can get involved in our society. We've been working to onboard a new member from the Reese Group. We have a contract with an executive director that we contract out. This was something that was started with David Baum's presidency. And we have a new executive director whose name is Kaylee Walters. She started a few weeks ago, and she joined us on Friday. Hopefully some of you got to meet her at the opening reception, but she's terrific, and we've worked a lot to kind of get her integrated into what we do here at SSB. A big part of what we've done this year, though, has been responding. So we've been responding to threats on science, but we've also been responding to threats that really affect the scientists who are doing the work. So along with Carol Boggs and Dave and Dan, we've been working together to try and be a strong voice for science and for scientists. So with that, I just want to remind all of us to please participate in a survey that's underway. All of the scientific societies whose logos are shown here, we're all going to be participating in a survey that assesses the impacts of US federal and state policies on science. The organizers of the survey are looking for responses by July 15th, and the hope is that this anonymized, aggregated data would be used directly by Congress and by policymakers. So if you want to have your voice heard, if you know other people who have stories, who have been impacted, please encourage them to contribute to this survey. For those of you who are SSB members, you should have already received the survey, but you also will receive a reminder on Monday. So check your email, check your spam folders. I want to give a plug as well to the Global Assistance Membership Program. This is something that Corrie Moreau and others last year really got off the ground. This is 2 new discounted membership categories for students, researchers, and educators in over 152 countries and territories that will give up to 100% discount for membership to SSB. So if you in this room feel like you would benefit from this program, please apply. And if you know others, your collaborators and colleagues who would benefit from this program, please encourage them to apply. I'd like to give a big shout out and many, many thanks to Isabelle Saint-Martin and Bob Thompson. They have worked along with OUP and Andy Seagram, our new rep for Oxford University Press, over the last year to 18 months to really get our flagship journal back on a kind of regular timeline and appropriate publishing schedule. There was a bit of a backlog, and Isabel and Bob have worked tirelessly to really kind of get us back on track, get into a regular publishing schedule, while still maintaining the high quality that we expect from our flagship journal and the intellectual rigor that exists for our flagship journal. So if you have articles that you think relate to systematic biology, phylogenetic methodology, please do still continue to submit to this journal, and hopefully you'll see the quick turnaround time that we all expect and know and love from our journal. We also have, thanks to the tireless work of Brian Karstens, the Bulletin of SSB. This is an open access only journal for our society. It is free for members to both read and publish in. So if you have work that you think may not be appropriate for systematic biology, but you think systematists should read it, or if you have work that you want to have openly accessible to the globe, please consider this other option to publish and keep your work in our home here at SSB. I'd like to give a shout out to April Wright, Communications Director Extraordinaire, who has been keeping the information flowing fast and furious through newsletters, email updates, but also on social media. So we had some discussion at the council meeting on Friday, and we are going to be archiving and discontinuing our Twitter or X account, but you can follow us on Facebook and Blue Sky, and often this is the fastest way to get information about what's happening right now in the society. So thanks to April for keeping us all informed, and if you have interesting ideas or news or updates, you can also contact the communications director so that can be shared with our broader group. I'll turn now to some awards. So we have several awards in our society, and it's really part of our mission. Part of our shared values are to invest in our members, invest in research, invest in science, and put our hard-earned systematic biology dollars right back into our society. And we do this through a variety of awards, the Ernst Mayr, Early Systematist, Graduate Student Research, Mini Arts, Publisher's Best Papers, and President's Lifetime Achievement Awards. And when you look at where we spend our money, we are actually spending a good chunk towards supporting this mission. So in 2024, we gave away over $140,000 in awards. And as you can see from the orange part of this pie chart, a big number of that was for Graduate Student Research Awards. And this was administered, those awards were administered by Becky Chung. So a big round of applause for Becky Chung for getting our awards distributed on a regular schedule to the people who were so deserving. Becky also organized the Ernst Mayr Award this year. For those of you who tuned in, there were some amazing talks. These are the finalists for the Ernst Mayr Awards this year for 2025. Really cutting edge, often the most curated and excellent slides of the conference, I think, are actually the Mayr Awards, students who put a lot of effort and time into making sure that they do good science communication, that their message is conveyed. So congratulations to all the award finalists. And a big thanks to all the folks who do the work to make sure that such an event can take place. So a thank you to all of the award judges, whose names are shown here on the slide. Thank you for working with Becky to make sure that there was enough people to see the talks, to judge the talks, and to give great assessment. And without further ado, I'd like to welcome Becky Chung to the stage. We have two 2025 Ernst Mayr awardees, Spencer Bab-Bernanke. If you're in the room, please come to the stage. And Zach Heipel, if you're in the room, please come to the stage. So Spencer Bab-Bernanke comes to us from Louisiana State University, and they will be speaking tomorrow at 12:15 in Athena AB on discordant topology, concordant chronology, coevolutionary history of fungi and their mammal hosts. Congratulations, Spencer. Zach Heipel comes to us from Scripps Institute of Oceanography, and they'll be speaking on Tuesday, June 24th at 11:45 a.m. in Parthenon 1 on the evolutionary dynamics of extreme tubular snout elongation in deep sea fishes. Congratulations. I have some news from Isabel and Bob about the Publishers Award for Excellence in Systematic Research. They wanted to let us know that they received over 400 manuscripts for publication in systematic biology for the year 2024, and they published less than 80 of these. So Isabel notes that it was a really amazing feat to have graduate students kind of rise to the level of this award. The awards was chosen based on the research that they felt was the most creative and likely to have a lasting impact on our discipline of systematics. The award comes with a prize of $500, which I do not have in my pocket here, so don't come up for the money, but you'll be receiving this shortly. The first award goes to Xiaozhu Peng, who I don't believe is here, but let's give a round of applause. And their paper focused on detection of ghost introgression. The next award goes to Rong Zhang. Another round of applause. And their work focused on fast Bayesian inference of data that is continuous characters. Now, we also participate along with the Tri-Societies in the IDEA Award. Jeremy's talk already happened on Friday, but how amazing was Jeremy Yoder's talk on Friday? It is now available to be seen online on the YouTube channel. And another round of applause to Jeremy for being an IDEA Award winner. So thanks to Elizabeth Jockish, social coordinator extraordinaire, who has found the Little Kings Shuffle Club as the venue for our informal mixer. This will be taking place tomorrow night from 7:30 to 10 p.m. I was just by there last night because there was a death metal band that was playing, and it was very good. I don't think there's going to be death metal tomorrow night, unfortunately, but we'll be meeting outdoors, weather permitting, and hopefully you can tell everyone you know and join us there. And so now it is my immense pleasure and honor to introduce the SSB Presidential Address, which annually is given by our past president, unlike for the other societies. And this year, our past president is Corrie Moreau. Dr. Corrie Moreau and I met first in 2006 at the Stony Brook meeting. And from the very first second that I met Corrie, she made me feel like I belong here. And over 20 years of friendship and colleagueship, I've witnessed time and again how Corrie makes space for others. She speaks out for the hardest initial in that forbidden acronym, the E, which is equity. And her leadership for SSB has been truly strong. She has advocated for all of us here, but also for all of those who are not in this room, but who should be. Dr. Moreau earned her Bachelor of Science and her Master's of Science from San Francisco State, and she earned her PhD in evolutionary biology from Harvard University, followed by a Miller Fellowship at UC Berkeley. She's now the Senior Associate Dean of Cornell's Ag and Life Sciences College and an endowed professor of arthropod systematics. Anyone who knows Corrie knows that she has ever been a strong voice for collections and the importance of biological collections, and she is the director and head curator of the Cornell University Insect Collection. Corrie is a fellow of many different prestigious organizations, including the Royal Entomological Society, the Entomological Society of America, the AAAS, and is a Kavli Fellow of the National Academy of Sciences. Corrie's work focuses on ants and their symbiotic bacteria, and she has published over 125 papers. But did you know that one of those is on dragonflies? So we can still get her back to odonatology, it's not too late. And without further ado, although I think there's no dragonflies in this talk, I welcome Corrie to the stage to talk about an inordinate fondness for ants, what evolution has taught me about change, resilience, and communication. So Maestro, Corrie.
Jessica, I almost cried. Come on, I gotta get a little trombone shorty going for the morning. Come on, everyone. Thank you. So I am so thrilled to be here today. I'm deeply honored to be up on this stage. Sorry, Jessica got me a little choked up. I never imagined I could be on this stage in 2006 when I met Jessica. And to know that she'll be here next year is just phenomenal. So I was thinking about, sort of the path that brings you to somewhere like being on this stage. And it led me to think about this idea of standing on the shoulders of giants, right? We've all heard it. We think about it a lot in science. And this phrase was first popularized by Isaac Newton in 1675 in a letter in which he said, "If I've seen further than others, it is by standing on the shoulders of giants." And I think many of us think about those that have come before us and both contributed to the scientific literature, but also those that have directly mentored us. So I want to spend a moment talking about one of those greats in evolutionary biology, J.B.S. Haldane. And Haldane had a lot of contributions to evolutionary biology which have been very long-lasting. One, he came up with this idea of this primordial soup hypothesis, which really is the foundation of the chemical origin of life. He established gene maps for hemophilia and color blindness on the X chromosome. He codified Haldane's rule, right, where he demonstrated that sterility is really related to the heterogametic sex in hybrid species. And he correctly proposed that sickle cell anemia disease conferred immunity to malaria. And he also coined cloning, and ectogenesis. So really you can see that he had many lasting contributions to the field of evolutionary biology. But I want to take a pause for a moment and sort of step back to sort of living in an organismal biology world and think about biological species diversity. We all know that the world is incredibly diverse and full of biological life, but if we look at the chart on the left, we see that almost all species that have names from scientists are actually insects. As an entomologist, I'm not surprised. Now, if we sort of look at the image on the right, we can see that almost among the insects, the coleoptera, or the beetles, are the most diverse insects in the world. So returning to Haldane, being a leader in evolutionary biology at the time, he was once asked by a group of religious leaders, what can be inferred about the mind of the Creator from the works of his creation? And Haldane replied, an inordinate fondness for beetles. Well, of course, I love beetles, but as most of you know, I'm actually much more in love with ants. So fast forward a few years, and Bob May was thinking about the same question. And he said, yeah, species diversity is incredibly important, but what about abundance? Let's look at what's around today on the planet. What's most numerically dominant and sort of taking over almost every terrestrial habitat? And he argued that what Haldane should have said is we have an inordinate fondness for ants. So I didn't know any scientists growing up. I'm a first-generation college student. I grew up in low-income housing in Louisiana, hence the shout-out to Trombone Shorty, my New Orleans heritage. But I loved science, and I loved ants from a very young age. And this led me to thinking about what allowed me to sort of excel in the field of evolutionary biology and entomology. And it really was both standing on, but also alongside the shoulders of giants. Some of these people came along long before I was ever around, like William Morton Wheeler and Eleanor Lowenthal. But then, of course, my direct mentors were Nomi Pierce, Scott Edwards, and E.O. Wilson. And they really helped me sort of form into the scientist I am today. But it's not just those that you sort of are standing on the shoulders of, it's those you stand alongside. And I'm fortunate to have 5 of my closest evolutionary biology friends here in the audience today. These people have pushed me to think about science in new and rigorous ways. They've helped me sort of contextualize how I think about our role in society. And so if you see any one of them today, give them a big knuckle bump for me because they've been really instrumental in my development. But it's also about paying it forward. So I've been really fortunate to be able to work alongside a whole bunch of amazing undergraduates, graduate students, technicians, postdocs, visiting scholars, and that has been probably the biggest joy in my academic career, is being able to work alongside and help mentor the next generation. I'm an organismal biologist, and when I was in graduate school, there was this big debate, it's been long going, I'm well aware, of whether question-driven biology or organismal-driven biology is the best, or the most rigorous, or whatever sort of way you want to contextualize it. So I thought, today is the day to give us the definitive answer. So where do we get all of our best definitive answers today? I called up ChatGPT. And I asked it, question-driven biology versus organismal biology. And it said that they're not mutually exclusive, but they represent different orientations or approaches to biological research. And so, of course, like every other person who's given a plenary at this conference this year, I thought I had to show you an obligatory XKCG cartoon. So the first panel is, I'm having second thoughts about grad school and could use some advice. And I think all of us have felt that way. Consider the fire ant. When there's a flood, fire ants survive by joining together into giant floating rafts. Huh? What lesson am I supposed to take from that? Ants are so cool. Well, you're not big on metaphors, are you? I'm big on ants. And I swear, when this cartoon came out, first off, a lot of people sent it to me. But secondly, I was like, I think that was written about me. All right, so as an organismal biologist obsessed with the little things that run the world, I think a lot about where we find biological diversity and why it's distributed where it is. And so ants are a classic example of the latitudinal species diversity gradient, where we have the highest diversity centered around the tropics. So on this map, the warmer parts of the map in red and oranges are where we have the most species of ants. And as you can see, as we move towards the poles, we're decreasing that species richness into those blues and greens. And so being an ant biologist, this means that I've spent a lot of time in the parts of the world where you find the most ants. And I've been really fortunate to be able to go and collect ants in rainforests around the world. Now, doing tropical field work is spectacular. I've absolutely loved every experience I've had. The picture in the top right that looks like a little spider in a web, that's actually me driving myself around with a remote canopy access system to collect ants from the top of the rainforest canopy in South America. And while tropical biology is rewarding and fun, it doesn't come without its pitfalls and haphazard situations. So the picture in the bottom right is actually a tick that I inadvertently smuggled home in my nose from Uganda, Africa, and it's the second known specimen of a whole new genus of ticks. One of the things I absolutely love, though, is taking students to the field and letting them have their aha moments, letting them develop their own research questions from being out in nature, and having them explore ideas and witness things and observe things. And so this is something I find really rewarding. But I do want to spend a moment talking about the elephant in the room. When I knew I had to give this talk a year ago, I had all these fancy ideas of like what I would mention and how I would address this. I even actually, during the last evolution meeting, had a group meeting. My entire lab was there. There were about 10 of us. We sat down and we brainstormed what I could talk about, how we would do it. We'd even come up with this idea they would all come up on stage, and then everything shifted. And I felt like that wasn't the talk I should give anymore. So I know that we're all in these confusing, difficult, troubling, and if you're looking for a drinking game word, unprecedented times. We know that there's assault on diversity, equity, and inclusion work. I've been a DEI dean for the last 3 years at Cornell, so I've felt it acutely. We know that there's assaults on higher education itself, assaults on science. There's mistrust of science and scientists. There's been massive cuts to research funding and travel restrictions that are inhibiting our colleagues from being able to attend this conference, but will have long-term effects on our ability to do the collaborative science that actually helps propel our research forward. But I would argue that as scientists, we're all trained skeptics, and we know how to debunk hypotheses. And this is a skill we need to use now. So when I knew that this was the talk I was going to give, I had a conversation with 3 of the graduate students in my lab, and I said, if you were sitting in the audience, what would you want to hear? And I thought for sure they were going to tell me, like, make sure you address this, and don't forget that. And what they said was they wanted to hear something that gave them hope and a little science. So that's what I'm going to do with the rest of the talk. So I was thinking about what ants have taught me through time, and I sort of put it in these bins or categories of change, resilience, and communication. So first, let's talk about change. Evolution is change, right? It's just change through time. And we know that we can go out into the natural world and understand how we ended up on all the biological diversity we see by contextualizing it in this framework. In addition, nature is incredibly resilient. We see it adapting to both the natural world, but even work by Shane Campbell Staton has shown us that it's adapting to the Anthropocene, the world that we're creating. But guess what? We're also resilient. And we have opportunities to help change the future. I've had great opportunities like being an invited speaker for the March for Science alongside Tracy Heath. And I've been out demonstrating with fantastic people, including my partner, Christophe Duplay. So now I'll talk a little bit about some of the research we do in my lab. First is on understanding the evolution of ant-plant interactions through time. So ants and flowering plants seem to have a very close evolutionary relationship, and that might actually come as a bit of surprise, because ants are actually not pollinators. So it begs the question, why would we expect to see any linkage between the diversification of the flowering plants and the ants? Well, from the ant point of view, right, when the flowering plants diversified, it provided all these new niches for them to occupy and hunt for food resources. And in fact, Wilson and Holdobler in 2005 came up with their dynastic succession hypothesis, where they had posited that some lineages of ants, which we know have tight ecological relationships with plants now, would have benefited in evolutionary time from this explosion in flowering plants. In addition, we know that many ants have shifted their diets to having a much more plant-based diet. So maybe this actually helped facilitate it with the rise of the flowering plants. But from the plant's point of view, ants are also incredibly beneficial in the form of seed dispersal. Seed dispersal by ants has evolved at least 100 times independently across the flowering plant tree of life and is known from over 11,000 plant species. In fact, it's thought to be so important to plant evolution that it's considered potentially a key innovation to the success of plants. So what we wanted to try to understand is, although ants are not folivores, and they do incorporate plants into their diet, what is that relationship between ants and plants? Now, the way that ants feed on plants is often through things like extrafolianectaries, right? These little sugar water fountains that plants produce to attract ants to engage in defense against herbivores. Some plants even produce these specialized food bodies specifically to attract and nourish ants. And of course, we've already talked about eliosomes for seed dispersal. But most feeding on plants is actually happening indirectly through tending sap-sucking insects like these aphids you see here. So to understand this question, the first thing we need to do was infer a very large phylogeny for the ants. And we learned a lot from that, but what I wanted to understand was sort of what was the pace or tempo of evolution for ants? And what we saw is that about 200 million years ago, ants were just getting kicked off. There were very few species around. And then right around 100 million years ago, we see this sharp increase in the diversification of ants. So we wanted to understand what could explain this pattern. Could it just be chance? Could it be some artifact of the way that we're looking at our data? So we looked at a set of models, and what we found is that the model that says we have two different breakpoints. There's one sort of pace of evolution before 100 million years ago, and then a drastically different pace around 100 million years ago, after them. And so when we look to the botanical literature, what we see is this almost completely lines up when the botanists believe that the flowering plants really exploded across the planet, suggesting that ants and angiosperms are highly correlated in their diversification. So then we wanted to understand, how are ants and plants potentially linked? I already talked to you about all of these structures that plants have evolved specifically to attract their defensive arthropods like ants. So we wanted to know, what were the age of those structures and when did ants begin to utilize these structures? Was there some sort of evolutionary link between these different patterns or traits? So the first thing we did is we took advantage of a very large plant phylogeny that has almost 11,000 plant genera. We then went in and coded all the tips for whether they had associations with ant-associated structures like extrafloanectaries, eliosomes, and then domatia. So domatia are those structures you see in the yellow box, which are these specialized structures that plants produce specifically to house their defensive arthropods so that they have a place to store their food and rear their young. Then we inferred a very large ant phylogeny that has over 1,700 tips of species, and then again did ancestral state reconstruction on those traits that are correlated with their utilization of plants. So before I walk you through what I found, I want to remind you that the earliest lineages of ants were ground nesting, they were ground foraging, and they were predatory. We know this not just because the earliest lineages of ants retain these characters, but also due to their closest sister relatives and from the fossil record. And then what we see is the first transition is that ants begin to forage arborally, right? They're going up into the canopy and hunting for food sources. Next, we see that ants begin to include plants into their diets. This is likely through things like licking plant wounds initially. And next, we see that plants begin to respond. We see the evolution of extrafloanectaries, right? Now the plants have enough ants running around on them that it's advantageous of them to attract ants to stay on that plant to deter herbivores. Next, we see now ants begin to nest arborally. They're no longer even going down to the forest floor. They're living their entire lives in the canopy. And again, the plants respond through the evolution of eliosomes. They're taking advantage of ants to disperse their seeds. And lastly, we see that most complicated structure, the evolution of the domatia, to house their defensive arthropods. So essentially what we were able to demonstrate is that ants were utilizing plants long before plants had evolved these specialized ant-associated structures. In addition, we see this incremental sequence of evolution of these traits that both ants are relying on plants, right? They're going from ground nesting and ground foraging and being predatory to then foraging arborally, incorporating plants into their diet, and then nesting arborally. And intermittently, we're seeing that plants are responding by evolving structures specifically to engage with ants. We continue the work to try to understand the ant tree of life in even higher resolution. We have a paper that's just been accepted in systematic biology. It's available as a preprint if you're interested in learning more about ant evolution. Hopefully most of you were able to see Megan Barkdale's talk yesterday. She gave a fantastic presentation. And we've been working on a project trying to understand the evolution of soldiers within the genus Cephalotes, or the turtle ants. And what we've been able to demonstrate is not only did the soldiers evolve 3 times independently, but really what we wanted to try to understand was, how do you take a single genome and make such distinct morphs? So within the turtle ants, of course there's a queen, but among their workers, there's 2 sizes or shapes. The first is the minor worker, who's responsible for going out and foraging for food and caring for the young. And then we have this large soldier with this exaggerated head disc. Its sole role is for defense, blocking the nest entrance in which they live. So what we did is a 2-pronged approach. We used a comparative genomics approach to look at selection on genes and gene regulation across pairs of species that both have a soldier and do not. We then took a developmental transcriptomics approach to try to understand what gets turned on when you're making a soldier. We asked the question, are we seeing selection on genes? Are we seeing selection on regulation? Or are we seeing the emergence of new genes that is allowing the evolution of these exaggerated soldiers? And what we're able to show is that when we looked at differential expression of genes, they are associated with the presence of a soldier morph, and that genes involved in this morph differentiation are actually disproportionately ancient, and that's the work that Megan focused on yesterday, so hopefully you were able to see it. So what we're able to show is that it's not, in fact, selection on genes that's allowing them to evolve these soldiers. We do see that it's likely selection on regulation of those genes, but it's not the emergence of new genes. But in doing this work, we're actually able to identify a novel signaling pathway that likely allows the formation of this soldier from the same genome that also creates the queen and the worker. So stay tuned, we're getting ready to submit this if you want to know all the finer details. So just like the natural world, I would argue that we are capable of change. And I really love this quote by David Suzuki, where he said, "There are some things in the world we can't change: gravity, entropy, the speed of light, and our biological nature that requires clean air, clean water, clean soil, clean energy, and biodiversity for our health and well-being. Protecting the biosphere should be our highest priority, or else we sicken and die. Other things like capitalism, free enterprise, the economy, currency, the market, government, are not forces of nature. We invented them. They are not immutable, and we can change them." So now I'll spend a moment talking about resilience. How do we adapt in the face of change and challenge? So I was thinking about the ants again, and I was thinking about how they solved a really hard hurdle. How do you go from being entirely predatory to then being entirely herbivorous? We know that it's evolved at least 11 times independently across the group. And with the challenges of both shifting diet, but also in nutritional content, it begs the question, how do you gain enough nutrition when you've drastically changed your diet so significantly? So this is where my longtime collaborator, Jake Russell, and I first started exploring this question. There was not a lot known about the diversity of bacteria associated with ant guts at the time. So when we first started this project, I sort of felt like one of those early rainforest explorers that was just collecting everything you could see. And I will say this was quite observational, as it needed to be. It was exploratory. So the first thing we did is we dissected the gut of almost 400 individual ants from about 150 ant genera and just asked what's there. And not surprising, there was a lot of diversity. But what jumped out to us initially was that we see this enrichment in a group of bacteria called the Rhizobiales. Now this order might be familiar to you, especially if you work on plants, because it contains the rhizobia, which are these group of bacteria that are associated with the root nodules of leguminous plants, where in return for carbohydrates from the plant, these bacteria fix atmospheric nitrogen and provide that to the host. So we wanted to understand, why are we seeing a group of bacteria from within the same order within the guts of ants? In addition, we wanted to understand which ants were enriched for this group of bacteria. So this is where we turn to stable isotopes, and so what you're looking at is the ratio of heavy to light nitrogen as it moves up the food chain. Every one of those triangles or diamonds is an ant genus, And so what you see is from things that are incredibly herbivorous all the way through things that are highly predatory. That orange diamond that's all the way to the right are the army ants. And then on the y-axis, we plotted whether they had an association with rhizobiales. And what you'll see is that we're far more likely to find an association of this group of bacteria with ants that are feeding low on the trophic scale. But if you have a keen eye, you might have noticed there's a couple of those diamonds that are actually very low on the trophic scale and don't seem to have an association with rhizobiales at all. And those are the carpenter ants and their relatives. And this is one of the only group of ants that we actually knew much about their bacterial associations. And in fact, in this group of ants, the association is so tightly linked that the bacteria are sequestered into a specialized pouch called a bacteriocyte. Then they've co-diversified with their host. And in this case, we know that they upregulate the host's nutrition. So it appears if you're going to feed low on the trophic scale, you're likely to have an association with some kind of gut bacteria. What you're looking at here is a phylogeny of the bacteria, but color-coded and named by the host or environment from which they came. And what you'll notice is that almost all of those that come from ants form a very tight clade that we're calling the rhizobiales clade. In addition, if you dive into that clade, you'll see that those groups of bacteria are tracking their ant hosts. If we look at the clade in the bottom part of that red clade, we can see that cephalotes, the turtle ants, actually are grouping together the samples. In addition, they're sister to Procryptoceras, which is their closest relatives, suggesting there might be a long evolutionary history of this group of bacteria associated with their ant hosts. But if we flip it on its side and look at it from the other direction, what we see is there's at least 5 independent associations of this group of bacteria with their ant host, suggesting that there must be some amount of jumping between ant hosts. But again, all of these ants are in fact herbivorous. So thinking again about the fact that some ants have this specialized structure, the bacteriocyte, there are other ways in which ants are keeping bacteria within their gut. My colleague Yi Hu has been doing some beautiful work on ants within the genus Tetraponera, which have this bacterial pouch in which they hold their symbiotic bacteria. And then the groups that we work on actually just have them distributed across their gut lumen. We know quite a lot about how bacteria can contribute to their host's health, but what we wanted to do is spend a moment looking at the bacteria associated with the turtle ants, again, who have this sort of diverse bacterial community that's distributed across the digestive tract. So what we want to understand is, where do we find bacteria? Because if the bacteria are in fact upregulating the host nutrition, we'd expect to see some amount of tissue specificity. So what we did is we went in and carefully dissected out the crop, the midgut, and then further divided the hindgut into the ileum or large intestines and the rectum. We did this across many, many samples of turtle ants. We had both replicates from within species, and we actually sequenced the bacteria across about 25 species of turtle ants to look at their bacterial communities. Now, just to remind you, the crop is the social stomach within ants. We don't expect any digestion to occur there. This is essentially a holding chamber while they're out foraging for liquid resources so they can carry them back to the nest to regurgitate it to members of the colony. This is essentially the same function of a crop as we see in birds. It's a storage chamber where they can bring food back to the nest. Now, we know that digestion begins in the midgut, continues within the ileum, and then probably drops off quite a bit in the rectum. So the first thing we did is we went and carefully dissected all of those samples so that we could look at the bacteria within each of those segments. And what you're looking at here is what we found. So each individual bar is an individual tissue from a species of turtle ant. The identity or color of each of those tells us what bacteria we found. And the first thing you'll notice is that the crop is incredibly diverse. Almost every individual looks different from one another, but each individual sample is more or less dominated by a single group of bacteria. Once we move into the midgut, it's almost entirely dominated by one group of bacteria, the Opitutales. Now, this is striking because these ants are found from the southern United States through Central America and well into South America. And we have sampling across that distribution. Now, once we move into the ileum, although there's some variation, it's actually quite stable across all of those samples, suggesting that these are also likely beneficial and have a long-term association with the host. And the rectum seems to recapitulate what's upstream and may actually still have some function. But it's not just the identity or where we're finding these bacteria, it's also the quantity that seems incredibly important. When we use quantitative PCR to measure how much bacteria are in each of these structures, you can see within the crop there's almost nothing. In fact, we find only about an average of 5,000 bacteria within this tissue across all the samples. When we move into the midgut, we see a huge abundance of bacteria within this structure, suggesting that we're likely seeing a lot of function happening there. We see it retained within the ileum, and then again it drops off once we get to the rectum. Now these are all indirect lines of evidence that these bacteria are likely important for the nutrition of the host. So we wanted to investigate further, could we actually demonstrate that there's a function? So what we did is we went in and sequenced the genomes when we were able to culture these outside of their ant host. It turns out that that's pretty difficult. Then we've also turned to metagenomics and sequenced all the bacteria within the gut. And what we've been able to demonstrate is that these bacteria retain all the genes to synthesize amino acids, so potentially upregulating their host's nutrition. But we need to go one step further and demonstrate that they're providing them to the host. Because again, these bacteria, since they're living in a community, could be synthesizing these to share with other members as a common good. So what we did is a series, apologies on that. We did a series of experiments where we took colonies that are genetically identical, split them in two, gave half antibiotics and half not, and then we enriched the diet of some with labeled urea. And what we were able to show is that those colonies in which we had left the bacterial communities intact, we had knocked them out with antibiotics or enriched their diet with urea, for the essential amino acids, they look identical. Now, we didn't measure these in the gut because, again, these might be produced as a common good for sharing between bacteria. We actually measured this labeled isotope in the hemolymph, or the blood of the ant within the leg. But when we leave the bacterial communities intact and enrich their diet with labeled urea, we see a huge increase in the amount of these essential amino acids that are in fact being provided to the host. And what this shows us is that, at least within the turtle ants, that the gut bacteria are synthesizing these essential amino acids via nitrogen recycling and providing them to the host. So my colleague Jacob Russell is actually giving a talk today. I hope that you all can make it at 11 o'clock this morning to learn more about how insects are utilizing bacteria to enrich diets. So this leads us to the question, what are they in fact using all these nitrogenous materials for? And it led me to think about, how do ants form a tough cuticle? And this is a nice little aside of being an organismal biologist and sort of a thought to, all of our future high-level researchers in the room, that I never thought I would study the insect cuticle. But as I sat at my microscope carefully dissecting thousands of ants, what I had noticed is that some ants were harder to break through than others. You've known it, if you tried to pick up an ant, some you can squish almost immediately, and some almost are like spines to your finger. And what I noticed is the ants that were the toughest to break through were either predatory or were herbivorous, and we were documenting that they had gut-associated bacteria. So it led me to ask the question, could the bacteria be contributing to the formation of that cuticle? So, as I've already mentioned before, we know that these ants have these nitrogen recycling bacteria in their guts. And one of the things that we were able to demonstrate is that these bacteria can synthesize phenylalanine and tyrosine, which are the precursors to formation of cuticle. So this asks the question, could they be contributing to the formation of that tough or rigid cuticle? So when we started this work, there had been some investigations to whether bacteria could be contributing to the formation of cuticles within 2 species of beetles. When we did this work, there were 5 papers published. And what they were able to demonstrate is that when you use antibiotics to knock out their symbiotic bacteria, the beetles become pale in color, and that their cuticle becomes significantly less thick. And in one of those cases, because they had sequenced the genome of the symbiont, they were able to demonstrate that the symbiont had all of the precursor steps that were necessary to synthesize the components that contribute to the cuticle, minus the last step in which the host had to contribute. So we wanted to understand, what are the specific cuticular components that are involved, right? We know that it could be things like the protein, the linker, the chitin, or even the melanin. We wanted to know, are there other potential pathways besides the one they see within the beetle? And are these mechanisms conserved across many species? So what we did is, again, return to our old friend the turtle ant, took colonies and divided them in two. We gave some antibiotics, right, just as we had done before, and labeled urea so we could track that component enrichment throughout their diets. And then we let the experiments run for several months. And the reason we needed to do this is we needed individuals to go all the way through development and incorporate these experimental manipulations into the formation of their cuticle. So when we took these colonies, we took all the workers that were around, because we need workers to care for the young. The young will not eat without having workers to care for them. Painted them, and then we knew which ones had emerged since the experiment. We then went in and used liquid state NMR to look at different components of isotopes within the system. We also used scanning electron microscopy to look at anatomical features of the cuticle itself. And this is where I partner with some analytical chemists to say, what do we in fact see when we provide these high-level antibiotics to knock out their symbionts? And what we see is we see a huge decrease in those precursors to the formation of the tanning process when we give them antibiotics. And this is exactly what we had seen before. Now, to understand what we should expect to see on NMR, so just to remind you, NMR is an analytical chemistry technique that allows you to trace a single isotope in a sample. We turn to your favorite chemical supply company, and you can buy a jar of pure chitin. And so what you'll see is this is exactly where we expect to see the chemical shift of chitin within our samples. Then we took some ants in which we had not done any manipulations and scraped out all their insides and ground up their cuticle. And the first thing you'll notice is that our NMR spectra is much lower, and that's because we have far less starting material. But you'll also notice that it's much wider, and that's because it's not pure chitin. Chitin, we know, has other materials embedded within it, especially proteins. Now, when we give these ants antibiotics and we knock out their bacterial communities, this is the signature that we find. So again, we see that peak of chitin and proteins together, but we also see peaks in the guanine arm of arginine, which is responsible for linking the chitin with other materials. But when we leave their bacterial communities intact, we see a huge enrichment of all of the components of the cuticle. We see an enrichment of both chitin itself, we see an enrichment of the guanine arm of arginine, of the linkers that are responsible of joining the chitin with the proteins, as well as the imidazole arm of histidine. So what this tells us is the bacteria are in fact responsible for almost every component of the formation of the cuticle. Now, when we use scanning electron microscopy to look at characteristics of the cuticle, these are not to scale, but one is about 50% less thick in the ants in which we knocked out their bacterial communities. So we're seeing just what they saw within those beetles, right? A shrinkage of the formation of the cuticle or the contributions to the cuticle. But unlike in the beetles, we actually see no sign of a decrease in melanization. And we actually knew this. We've been getting antibiotics to these ants for a very long time in lots of different kinds of experiments and never seen any depigmentation. So if you think of the tanning process, it can go down one of two arms, either the melanization pathway or the sclerotization pathway. And what we're seeing is there's a huge impact on the sclerotization pathway, but not on the melanization pathway. So this allowed us to identify what is likely the biosynthetic pathway that's leading to the formation of the cuticle that's being assisted with their symbiotic bacteria. And so what we're able to demonstrate is that ants that live on the extremes of diet, and today I've only presented work on the herbivorous diet, but we've also collaborated with Jake Russell and his team on some predatory species, and we've shown that when you get to extreme diets, you're often relying on bacteria to enrich the nutrient suppressed diets that you're consuming. So now I want to take a moment and highlight some of the work that a graduate student's doing in my lab that I think really does address this questions of nature and resilience. And she's asking, how do indigenous fire practices impact biodiversity? So this is work that's being led by Cheyenne Rubin Thomas. She's actually not here because she's in the field right now. And she's testing the impact of indigenous fire practices on insect diversity. So cultural fire, or sometimes called good fire, is this indigenous practice that communities have used for millennia that promotes fire-adapted and fire-dependent ecosystems. And this is an example of one of the sites that she went and surveyed last year, where she looked at this ideal site where they had done a burn, and just 3 months later, you can see how resilient that community has come back. And this allows those communities to rely on things that they've traditionally made in exactly the right form for millennia. And she's actually comparing that work to fire practices that have been incorporated by US-led agencies to ask the question, if they're using these good fire land management practices, what is the impact compared to these indigenous-led fire practices on biodiversity? And so she's actually sampling sites in both of these places, both right after a fire, in places where fire's not being used, and in nearby natural areas. And what she's trying to understand is, does cultural fire increase species diversity for insects when we look at them? And then also, what happens to genetic variation? How resilient are those populations in each of these habitats? Now, like you and me, you're probably eager for the answer. We don't know yet. As I mentioned, she's currently right now out sampling. She's sampling both in her own nation, the Oneida Nation in Wisconsin, as well as the Iraq Nation in Northern California. So come back to Evolution next year and learn what she finds. So I like to think that, like the natural world, we're really capable of resilience, that all of us can respond, and even in these troubling times, there are lots of ways in which we can actually act to make change. Last, I want to spend a moment talking about the importance of communicating science, because I think that we have all learned in the last 6 months how much our message hasn't necessarily been landing. So one of the ways I think that as a university professor I have the ability to make a huge impact is in the classroom. So in 2020 I started teaching a class called Diversity, Equity, Inclusion, the Science Behind Bias in STEM. And really what we were trying to do was have students understand, and interestingly, every time I've taught the class, we've had undergraduates, graduate students, postdocs, and faculty enroll in the class. What we want to understand is what's the impact, both historically and contemporarily, to the way that we've been biasing who's included in our research fields. So what we do in the class is we actually start with the semester with some community best practices. For many individuals, this is the first time they're engaging with material like this. So we're setting the stage on how do you have difficult conversations, but we're also introducing a lot of important things they need to consider as they move through the semester, things like intersectionality and privilege. We then move into the sort of modern published scientific literature about what does the literature say about what we know about who's being excluded and why, and what the impacts of that are. We then break into small groups and discuss it, and at the end of the semester, every student in the class completes a reflection or dives into a topic to investigate it further so that they can share how they plan to contribute to making equitable and inclusive spaces moving forward. We actually published a paper on this. I got to do this with a whole amazing group of individuals. Drea Darby, Amelia Demaray, Lena Acelia Hernandez, and Claire Meters. And here are some of the topics that we address when we teach the course. We start with a historical perspective on racism in STEM. We then talk about what does the data say against women in STEM, LGBTQIA individuals in STEM, people of color, people with disabilities, first generation and low income students, people with disabilities, and just trying to understand what does the data actually inform and tell us. We then talk a little bit about the impacts of systematic racism and bias and exclusion in STEM, and then we talk a little bit about what progress we have made. And we end out the semester identifying actionable steps that each of us individually and collectively can do as we move forward. This is a framework that we developed for the class. Really what we're doing is we're addressing these issues of racism and white supremacy, how bias, eugenics, and human experimentation have all led into how we think about science and how it's formed the institutions that we're all part of now. We then have this ability in the seminar class to do community learning, identity and positioning, understanding the impacts of those historical legacies, and how we can make action and change, so that hopefully when students come out the other side and they're moving into the STEM community, that they understand how to create flexible pathways, they can achieve equity in STEM, and really address and recognize where racism, bias, and exclusion is coming from. One of the things in the paper that we created, and I've put this QR code up for folks who might want to teach a class like this. So part of the reason we wrote this paper is we kept getting asked, how do I teach a class like this? How can I morph it away from just STEM fields? What happens if I want to have a physics focus? So what we did is we wrote a paper on how to teach classes just like this. You're always welcome to. That QR code gives you not only the paper, but it links to a website where I post all of the syllabi for the courses that we've taught that include the full list of all the readings or media we've used in the class, as well as that first introductory PowerPoint presentation on how do you address difficult conversations, what are some of the things, the challenges that the students might encounter as they're moving through the semester. One thing I do want to highlight is I'm particularly excited that some version of our class is now taught at a lot of other institutions. We know that it's being taught at the University of California, San Diego, the University of Toronto, the University of Texas at Austin. Shout out to Kelly Zamudio right here. Clemson, UNAM in Mexico, California State Northridge. And if any of you ever end up teaching a version of this course, I'd love to know because I like to keep track of how many places this course is touching. So I also want to take a moment to focus on another graduate student in my lab who's really spearheading some very interesting work trying to directly address how historical racism is having an impact on biodiversity in cities. And this is the work of Sylvana Ross, and what she's trying to understand is how historical practices of racial segregation in the form of redlining in cities in the United States might be impacting the natural world. So redlining is this initiative in which government agencies and financial institutions partnered to designate neighborhoods as high-risk areas for investment. And what this means is they went around about 200 cities in the United States and then designated each part of the city as being the best, is designated in green, and those are called A-graded neighborhoods. Not surprisingly, they're almost predominantly white and very wealthy. Next, we have blue, which are still desirable, or B-rated neighborhoods. These are often still white, but often lower income. Then we have what are called the yellow, which are definitely declining, or called C neighborhoods. These are often recent immigrant neighborhoods. And lastly, we have the red, or the D-graded neighborhoods, which are considered hazardous and host most of our Black and other marginalized populations within these cities. Now, the implications for this is this practice resulted in limited financial resources for high-risk labeled neighborhoods. And what this did is essentially perpetuated further racial segregation, economic disparities, and prevented the accumulation of intergenerational wealth. So we can still see the signatures of redlining in almost all of our US cities, even though this has been ruled illegal for almost 100 years. So what Sylvana's doing is she's going across both Philadelphia and Baltimore. I'm going to show you some of her Philadelphia preliminary results today. And sampling ants of a native ant species, Taponoma seseli, from each of these different neighborhoods, the A, B, C, and D graded neighborhoods, as well as nearby natural areas, to ask questions about what is their thermal tolerance. Because one of the things that we know is that the neighborhoods that are graded as D and C often are urban heat islands, have some of the most amount of concrete and the least amount of green, and also often have the most toxin pollutants in those neighborhoods. She's also asking questions about what's the differential of gene expression between these different populations in these different neighborhoods. And lastly, we're doing a whole bunch of genomic sequencing and then asking, do we see associations with some of these environmental variables with the species, with the individuals we're finding in these neighborhoods. So some of her preliminary results of the thermal data is showing that ants that she's collecting in those degraded or red neighborhoods have much higher thermal tolerance. We see that persisting in those C-graded neighborhoods, and that those ants that we're collecting in those A or the green neighborhoods look no different than the natural areas, suggesting that the ants really are responding to this change in the way that we're shaping our cities. Sylvana is highly committed to science communication. She's done it in a myriad of ways. Definitely she's one to watch out for. But just recently she was featured in a PBS series called The Bugs That Rule the World. If you haven't seen it, please go watch it. Not only is she involved in she's featured in all 4 episodes, They highlight her research in 2 episodes out of this series. Now, even if you don't necessarily want to watch it to promote Sylvana, please continue to watch things on PBS because they are also under assault from our current administration. So just like the natural world, I think that we're capable of evolving our ways of communication. One thing that I've been incredibly committed to throughout my career is trying to find ways to meet people where they are. I love being at a scientific seminar. I love talking to my colleagues with jargon. It's taken me a long time and a lot of training, intentional training, to try to figure out ways that I can get it out to the general audience. Sometimes it's things like going out on the field of the Chicago Cubs and bringing all of my science. Sometimes it's just being out on a museum floor to discuss with the visitors what's going on. I got to attend a K through 12 education expo that was for those students of that age, and they turned me into a science playing card. And I even have partnered with people like Emily Grassley from the Brain Scoop to create YouTube videos. This particular video has been viewed over 2 million times. Now, any of us who are scientists usually hate to watch ourselves or listen to ourselves on these kind of things. We're often afraid we're going to make mistakes, or that's not exactly accurate the way I conveyed it, or maybe that's a little not how I wanted to say it. And I will tell you that's true for every one of these things I've done. But the impact it's had positively, that most people don't see that one little thing that I could have said a little differently, is much more important than my colleagues who are like, hey, Corrie, you made a mistake when you said that one thing in that YouTube video. Yeah, I know. That's not who the audience is. So think about who your audiences are. Now I think it's really time for all of us, and I think that's what we've learned since January, is that each of us has to find ways to communicate our science. I would say be innovative and creative. All of us have different ways that we choose to be comfortable in the ways that we talk about the work we do. Team up with others that have been successful. If you want to create K through 12 materials to go into classrooms, you absolutely must consult educators. And that's because they have to conform to the learning standards that are required for them to meet to be in their classroom. So what you're seeing on the right is an example of that, where I partnered with some professional science educators to create resources that could go directly into the classroom because they conform to the next generation science standards. Also empower all the folks that you work with, the interns, the undergrads, the technicians, the graduate students, the postdocs. They're often almost of that age group. They often connect the best. Tell them that science communication is a valid part of their careers. It's not in addition to, it's complementary. And lastly, use any opportunity you can to talk about your science. I know sometimes with our families or our family friends, it can be difficult because there might be divides over their belief in some of the research we do, or even politics. You don't always have to hit things head on, right? You can just talk about your enthusiasm and slowly bring them there. You don't have to start with science is being cut and my grant was terminated. You might start more with why you do what you do and help them understand why that's so important first. As Jessica mentioned, I'm a huge fan of natural history collections. It's actually where I got my start. I've been in a natural history collection at every single stage of my academic career. I know just how critically important they are to both my science and all of you in the room. And if you think that you don't need a natural history museum, what's the name of the species you work on? Ask your colleagues the cells that they work on. What species do they come from? Do they have names? Right? These are all important. We need natural history museums. And they're often one of the first that are hit when funding is tight. We know that they educate millions of visitors every year, talk about being one of the best spokespeople for the science we do. They're often leaders in informal education. Almost every natural history collection has active researchers associated with them that are using the collections every day and making those available to other forms of scientists. They're research repositories for biodiversity, and they're resources for researchers in any field. And I know you might say, is that really true? But I will tell you, at the Cornell University Insect Collection, we've recently had material scientists come in to use our collections. They want to understand why insects don't shatter when raindrops hit them, so they can make materials better for planes. We've had chemists come in because they want to understand the evolution of pigments in insects, right? And those are people that historically you would not have thought of as coming to a natural history collection to investigate rigorous scientific questions. And I would argue that we all benefit from biological collections. Another place, and I think I'm preaching to the choir here, is that we all know the importance of scientific societies and their benefit not just to their own membership, to society more generally. Because they're often independent of funding agencies, they have the ability to stand up and say things collectively for us that as individuals we might be more vulnerable to. So please renew your memberships. One of the things that scientific societies do is they're often involved with advocacy, and they can have a unified voice. We've heard a lot about it at this conference, how all of our scientific societies have come together, but also partnered with others. They engage the public with the importance of our work, both sort of the general public, but as we've learned, even on the Hill. They're a great place for all of us to exchange ideas, to foster collaborations and advance our fields, and they bring us together, and there is strength in numbers. But I would also argue sometimes we all just need to protest. And I've been involved in activism since I was a very young person. It's something that I'm still engaged with today, sometimes on the forefront and ending up in jail for a night in San Francisco County Jail, sometimes just by showing up to protest, sometimes it's in the letter writing we do. There are many ways to protest and have your voice heard. But I also want to share that sometimes just doing your science is a form of resistance. And I share this because many of us in the audience, if you're an early career researcher, you might be feeling vulnerable. If you're from a minoritized identity, or if you're trans, you're incredibly susceptible right now. You might be thinking, I want to do something, but I probably can't. Just keep doing your science. Right now there's an attack on science itself. Us doing science is a form of resistance. For those of us who have privileged identities, please use that privilege and advocate for those who maybe can't be advocating for themselves right now. So I think we have a lot to learn from evolution. I've learned a lot about evolution from the ants that I study. Ant evolution was heavily shaped by the rise of the flowering plants. And in turn, plants have repeatedly evolved these specialized structures to interact with ants and have ants act as their bodyguards. Herbivory has evolved independently many times across the ants, and that's been facilitated by them bringing on these symbiotic gut bacteria to help enrich their diets. And we all need to contribute to making science and society more inclusive and equitable. So I've had the privilege of traveling around the world to pursue my passion for ant evolution, and I hope that all of us in this room get to continue exploring what we're curious about and what gets us so excited about the natural world. But at the same time, it's on all of us, no matter what our identities are, to make sure that we make the world more equitable and just for everyone. With that, I'd like to thank all of you for being here today. Thank you. I think we have just maybe 4 minutes for questions or thoughts or feedback. Welcome to any and all. Yes, Dan.
So, science detail question. Oh, perfect, thank you. The gut microbiome data that you showed where the crop had basically no microbes in it, what about the resources that they're ingesting into that crop? Are those microbe-free resources, or are they somehow actually eliminating microbes and have sort of a localized immune microbe suppression system in the crop?
Yeah, so what we think is happening is it's just reflecting what is the contamination on the food source they were holding last. So since they fill the crop and then they expel it all or pull some of it in digestion, it probably just reflects whatever is on the outside of what they've eaten last, which is why it's in such low abundance, why it's a single category, and why it's changing among all the individuals. So I think if, just like us, if you were to sample our stomach right after a meal, we'd see some bacteria there. It'd probably be dominated by whatever was on the food we ate last. But as we move through our digestive tract, that gets swamped out by the bacteria that are actually there for a functional reason. No questions, really? No hard-hitting ones? Come on. All right, well then you're all excused. Thanks, everyone.